All this is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. I hope you have not become sick of so many of the discussions about immune system. However, I thought this is important to clarify some of the immune system functions in light of some of the latest questions I'm getting from some medical students and doctors. And I thought it will be useful for us to know as well. At the end of the day, inflammatory diseases do involve the immune system, and we have to understand how our immune system works. Of course, I have done separate set of lectures about inflammation and how to modulate that, but this is an important corollary to those lectures. So let's start. So an overview of the immune system. You may have seen those diagrams with me where there is a macrophage, which is part of the innate arm, and that activates a T helper cell, and T helper 0 cell then becomes a T helper 1 or 2 cell, and then T helper 2 cell activates a B cell. T helper 1 activates a cytotoxic T cell. So that is an interesting diagram, and I've used that for the last 4 years as well. However, it leaves out certain mechanics of the immune system, which I think are important for us to understand. So let's start. This is about a pathogen. The pathogen could be extracellular. Many bacteria are extracellular. They do not enter the cell. They just sit outside and cause harm. Or intracellular. All viruses are intracellular. They get into the cell, steal cells' machinery, and use that for their own manufacturing of more viruses. In that process, they cause the cell to be destroyed. Also, cancers are intracellular pathologies. These are problems within the cell, and they are also handled in the same way as intracellular pathogens are handled. Some fungi also are intracellular. Some bacteria are intracellular as well. So the first concept to keep in mind as we go through the overview of the immune system is that pathogens can be extracellular or intracellular. They both are handled differently. The second important concept is the following that we have not touched on in our previous discussions. So imagine that we have an infection in our finger. There was some injury that caused some bacteria or viruses to enter our finger. So let's say these were bacteria. So now we have a tiny infection at the site of the injury. So let's say here is this pathogen, some bacteria or multiple bacteria, bacterium or bacteria. The first thing to keep in mind that the pathogen that is now developing and growing in that injury, causing infection, that pathogen will flow with lymphatic system. Lymphatic system is the system that picks up interstitial fluid, the fluid that is present between the cells, and especially that that part of the fluid that is more thick, has debris, has bigger chunks in it, has bacteria in it. Those bigger chunks cannot enter blood vessels, so they are sucked out of the tissue by the bigger channels called lymphatic channels. These lymphatic channels allow the pathogen to flow through them and enter a local lymph node. So this is important for our discussion because in the diagrams, we simply say that here is a pathogen and macrophage captured it and presented it to the acquired immune system. But it is important to notice that the T cells and B cells primarily live inside the lymph nodes. So pathogen has to reach there to be processed by these cells. So the second concept, the pathogen that is present somewhere will flow through the lymphatic channels and reach a lymph node or local lymph nodes. Next concept. In the area where the infection was, so let's say it was at the tip of the finger, over there are going to be innate arm immune system cells. The innate arm activates within the first 24, actually 10 hours to 24 hours. These cells are natural killer cells, neutrophils, macrophages, dendritic cells, and more cells. But these are, today we'll be discussing macrophages and dendritic cells. So here is a macrophage, and here is a dendritic cell. The difference between them, there is some difference in their origin, there is some difference in their function, but more importantly, macrophages will take care of the pathogen on the site of infection. So they, in the beginning of an infection, macrophages are the one that primarily try to handle the pathogen in that area. And then when the inflammation starts occurring, then some macrophages can actually go to lymph node. But mostly the macrophages stay in that area and help with the pathogen cleanup and help with the debris. So, of course, there is a fight going on with the pathogen. There are white blood cells that are present there. These white blood cells are going to be killed. Some of them are going to kill the pathogen. So now we have tissue damage. We have immune system cells present there, some of them dead and some of them fighting. And we have dead and fighting pathogens as well. That all together is present in that local area. So cleaning up of that area is macrophage's duty. Apologies for my throat. So here, imagine that this is a pathogen, this is a bacteria, and this bacterium is now connected by macrophages and dendritic cells. Macrophages and dendritic cells have on their surfaces something we say pattern recognition receptors, or PRR. The examples of these PRR is a bigger category that we use, and then under that category there are various kinds of receptors which are called toll-like receptors, and then there are other type of receptors as well. Their job, these receptors' job, is to recognize foreign agents. And they can also, excuse me, so recognizing the foreign agents is called pathogen-associated molecular patterns that are present on the pathogen, which are recognized by the dendritic cells and the macrophages. On the other hand, macrophages and dendritic cells can actually recognize broken tissue cells as well. When a tissue cell breaks, the cell would reveal its broken pieces. Mitochondria would come out of the cell, the broken nucleus would come out, the RNA would come out, some Golgi apparatus would spill out. All of these things, structures that are usually sealed inside the cell, are now spilling out. These structures are cumulatively called damage-associated molecular patterns, or DAMP. Of course, damage-associated molecular patterns. The cell got damaged, and these molecular patterns became revealed outside. This is like the cell's guts are spilled. So macrophages and dendritic cells can recognize pathogens through their pathogen-associated molecular patterns, receptors, and they can recognize damaged tissue through their damage-associated molecular pattern receptors. Once a macrophage finds, or a dendritic cell finds something offending, it will eat it up. It will phagocytose it. Phagocytose means to eat up something. So it will phagocytose that. So imagine that this is a macrophage. It has now eaten up that pathogen and internalized it. Once it has internalized it, it will then break it up into smaller pieces. Inside the macrophages, there are, and inside many of our cells, there are vesicles. Vesicles are simple, tiny bags. These vesicles contain lysosomal enzymes. They contain digestive enzymes. So once the macrophage has eaten up a pathogen, or eaten up a dead cell, or broken cell, or material from a cell, then the macrophage would fuse its digestive stomach, if you will, with this phagocyte, and the enzymes from this stomach will spill onto the captured pathogens and digest them and break them down. Once those pathogens are broken down, they are then loaded onto a protein which is called MHC2. major histocompatibility complex number 2, and pieces of those pathogens or broken cells are then presented as an antigen on these MHC2. So I used to talk about this in the past by saying, imagine that you have taken a bite of some food and then you chew it up, and then you take a piece of that and show it to the world and say, look what I'm eating. That is what a macrophage or a dendritic cell does. However, as I said before, macrophage will tend to stay in that area and continue to do this function. On the other hand, a dendritic cell is more capable of moving to the lymphatic channels and through that into the lymph nodes. So now imagine if my finger had the infection, there a macrophage had eaten up the pathogen and now it is just destroying it in that local area. And a dendritic cell had eaten up the pathogen as well, phagocytosed it. And now that dendritic cell is running along or flowing along the lymphatic system and will reach local lymph nodes. So in my case, if there is a infection in my finger, possibly those pathogens and the dendritic cells will flow to axillary lymph node or cervical lymph node or the lymph nodes near the opening, near the where the limb is attached. So far so good. The number third concept, macrophages and dendritic cells will phagocytose a pathogen, then they'll, especially the dendritic cells, will take those pathogens and go into the lymph node. So now think about the local lymph node. The lymph node has T cells in there, B cells in there, some macrophages in there, some dendritic follicular cells in there, the dendritic cells that are flowing in from outside, pathogens that are flowing in from outside as well. And the lymph node is a chemical lab. It is a lab where the pathogen pieces will be inspected, and then the immune system cells, the acquired arm, that is T cells and B cells, would decide what to do with them. Now remember, it is always the innate arm, that is the macrophages, dendritic cells, neutrophils, NK cells. They are the one who are the first responders. Neutrophils, for example, are the first responders in majority of the cases. They are called innate arm. They do not care what kind of pathogen is this. They just know it is foreign agent or a broken cell, and they just start attacking it. Innate arm's job is then to activate the acquired arm. We call it acquired arm because acquired arm has to acquire the knowledge of the pathogen and has to become activated by the innate arm. An acquired arm will not become activated for the first exposure of something. So imagine someone, a baby is born, and that baby is exposed to a pathogen, let's say streptococcus, for the first time ever in their life. This baby is, again, assuming that the baby has been of enough age that his or her immune system is now functioning. In the beginning, when they're born, their immune system is weak. It's not functioning very well. So imagine baby is, let's say, 2 years. Now their immune system is working. And the baby got exposed to streptococcus for the first time. In that case, the T cells or B cells will never become activated. First, the innate arm cells, the macrophages and dendritic cells, will have to pick up that pathogen, break it down, and bring it to the T cells and B cell, and bring that acquired knowledge and train them to, not train them, but to activate them to attack this pathogen. Once the T cells and B cells have attacked this pathogen, then they would develop memory cells. Those memory cells in the future with the re-exposure will become activated even if the innate arm is not helping them anymore. And they would start responding to that pathogen. So memory cells is a different discussion, but important to keep in mind. So here, the pathogen has flown or floated in the lymphatic channel to reach the lymph node. And some dendritic cells are arriving in the lymph node as well, which have captured the pathogen. Now, inside the lymph node, what's happening? Inside the lymph node, we have T cells that live there, B cells that live there. There are T helper cells. There are T cytotoxic cells. There are B cells. There are dendritic cells, follicular dendritic cells. There are macrophages coming from outside. There are dendritic cells coming from outside. And there are pathogens coming from outside. important thing to notice now is imagine that this is a pathogen that arrived in a lymph node. And here is a B cell that is bound to this pathogen. There may be other B cells that are just sitting there and they're not bound to it. They do not recognize this pathogen. So they're just sitting around. They're doing nothing. They're not bound. They're not looking for activation. They're just sitting there. Similarly, here is a cytotoxic T cell. And imagine that there is some infected cell which may be outside of the lymph node, and the cytotoxic T cell is bound to that infected cell. An infected cell, nucleated infected cell, can present the antigen, that is whatever has entered that cell. So either a viral thing or a bacterial component or a fungal component or even cancerous proteins in the early part of the cancer cells. Those antigens will be presented on the infected cells on MHC1 molecule. All nucleated cells present the antigen on MHC1 molecule. And here is a fun discussion that I had with a medical student a few days ago. That medical student said, we have two types of cells. We have professional antigen presenting cells. Professional antigen presenting cells are the cells, for example, we saw here, dendritic cells, macrophages, B cells as well. These cells can pick up a pathogen voluntarily, break it down and present it on their surface on MHC2. However, all nucleated cell who were not picking up pathogens voluntarily, the pathogen forcefully entered into these little cells. And now they are presenting it on MHC1. So a cytotoxic T cell will be bound to MHC1. And there is a very simple formula that we used to use, that MHC1 works with cytotoxic T cell, which is CD8 positive cell, we say. So 1 multiplied with 8 is 8. On the other hand, the cells that will work with MHC2 are helper T cells, and they work through MHC4. So 4 multiplied with 2 is 8 as well. So the binding has to be such that their multiplier is 8. This is just to remember that MHC2 binds with CD4 and MHC1 binds with CD8. CD8 are cytotoxic T cells, CD4 are helper T cells. So what is the takeaway from here? The takeaway is that there can be B cells that are bound to a pathogen, and there can be free B cells that are not bound to it in a lymph node. And then there can be T cells that are bound to an infected cell, and there can be T cells that are not bound to it. And you may have this question that why is it the case that some B cells are bound to the pathogen and others are not? The reason for that is the structure of their receptors. Every B cell or T cell has a slightly different receptor or binding region. And based on the structure of that region, one B cell might be able to bind to an area of the pathogen, while another B cell may not be able to. So this is just like this, that if you have a wrench that can pick up certain things, but it may not be able to pick up bigger molecules. Similarly, B cells are different from each other in the structure of their receptor. And so some B cells can bind to one pathogen, others can bind to another pathogen, and so on. Same is true for cytotoxic and helper T cells as well, that they can bind with one kind of antigen or another. So good. This is the next concept, whatever number it is, fourth concept, I think, that inside the lymph node, there are B cells that are free floating and just sitting there, and there are some that are bound. And then cytotoxic T cell as well, same. Some are free and some are bound. So now this little busy diagram, but I'll walk you through this, and this is a very important diagram. So starting from here, ignore all the rest, starting from here. So this is a dendritic cell. This dendritic cell had phagocytosed a pathogen. It had eaten up a pathogen, broken it up into smaller pieces, and is presenting one of those pieces here on its MHC2. On the other side now, we are talking about lymph node. On the other side, it is bound to a T helper 0 cell or naive T cell. So SL and TFRTR says there are multiple binding sites on the surface. Correct. So let's go and answer that question. Absolutely. So imagine that this is one bacterium. This bacterium has so many hundreds of patterns on it. And there can be many B cells or T cells that can bind to various patterns. So this is correct in the light of your question, SLNT, that there may not be just one B cell bound here, but there would be tons of B cells bound to various binding regions of this or various binding patterns of this pathogen, which we will call them antigens. So yes, you're correct. I am just trying to keep it simple. But this was definitely not to say that one pathogen can only have one B cell bound to it, or one infected cell can only have one T cell bound to it. Depends upon what is presented on the antigen, what is the shape of the antigen, and whatever T cell can bind with that shape will bind. And there could be multiple shapes expressed and multiple T cells bound here. Similarly, multiple B cells bound to the pathogen. Very good question. Similarly, multiple helper cells, CD4 cells bound to these structures. So very good question. You should get the award for today's lecture. Okay, so back here. The dendritic cell phagocytosed the pathogen, ran to the lymph node, presented a piece of pathogen and antigen on its MHC2. On the other hand, in the lymph nodes, now we have the cytotoxic T helper 0 cells or naive T cells. These T cells have no idea what their life is going to be like. So as soon as something comes their way to which they can bind, remember that T cells also have specific binding regions on their T cell receptors. And these binding regions are different amongst various T cells. And because of that, some T cells will be able to bind here and some will not. So that means there may be some other helper T cell that are just sitting around doing nothing. Even if they try to bind here, they were not able to. And they'll say, come on, leave it. I can't bind with this one. I'm just going to go back to my couch and sit there and be a couch potato. So back here, the T helper 0 cell is bound. Once this binding occurs, the CD4 molecule on the T cell, T helper cell, will bind with the MHC2. So this is a concept of co-stimulation in the immune system. The co-stimulation is that immune system does not want to be willy-nilly activated. So to really activate something, it is like if you ask someone to do something and they say, are you sure you want me to do it? And you say, yes, I'm sure. And then they call you over the phone and say, are you really sure you want me to do it? And you say, yes, I'm sure. And then they say, okay, send me an email that you want me to do it. And you say, okay, fine, I'll send you an email. That is double and triple checking within the immune system so that immune system doesn't become activated just by itself. So T helper 0 got bound through the T cell receptor. That is not sufficient. It also needs the CD4 molecule to attach to MHC2. That is also important. Then there is another molecule called PD1 on the T helper 0 that can bind with the PDL with the macrophage for further activation. Similarly, there are molecules CD28 and CD86. So there are multiple molecules. This binding area where these two cells are kind of handshaking with each other through multiple arms, this is called immune synapse. Synapse is really a very tiny gap between two things. So this is an immune synapse, and inside the synapse, all of these bindings are happening. Hugs and kisses are occurring. So, but this is still not enough. Now this T helper cell is ready to be activated, but it still needs more stimulation. And that stimulation is going to be in terms of some cytokines. You have to give it a shower. You have to bathe it in certain cytokines or chemicals, which are the last signal for it to become active. So when I talk with the medical students, I tell them that the cell now needs to be given a shower as well. And based on what is the chemical in the shower will decide how the cell would further react. So in this particular case, I made interleukin 12 to be the shower coming in. Where does the interleukin 12 come from? This dendritic cell or a macrophage can actually release interleukin 12 in the synapse. That interleukin 12 would bathe one side of the cell. Wherever the interleukin 12 is coming from, that side of the cell will become bathed. Now, please remember, there may be other T helper 0 cells here as well who would accidentally become exposed to the interleukin 12. But if these other T cells have not bound with some other cell, like another macrophage or dendritic cell, then you can bathe them as much as you want with IL-12 and they will not become active. For them to be activated, they will have to have all of these structures activated as well, and then interleukin 12 would activate them. So Margaret says, so it's like EPT testing essentially. Yes. Right? So SLNT FRTR has a very good question. The next one. So today you're going to have all the dollars earned from here. Okay, thanks for that. I also wonder, does the size of the pathogen determine how many binding sites are present? Absolutely. The bigger the pathogen size, the more molecular patterns and more amino acids present on its surface, which will make more patterns. So absolutely correct. So now the T helper 0 cell, in the presence of interleukin 12, coming from the macrophage or the dendritic cell, will become converted into T helper 1 cell. So now I want to clarify one more thing, and that is that in the diagrams, as I made over here, I made an arrow from T helper 0 to T helper 1 and said this will become T helper 1. It is actually right in place here that the T helper 0 will become T helper 1. It doesn't go somewhere else, but for illustration purposes, it is made separately to say now this has become an interleukin 1. T helper 1 cell. When it becomes a T helper 1 cell, T helper 1 cell performs a couple of actions. Number one, it releases interleukin 2. And for medical students out there, the way to remember this is that interleukin 12 showers on the T helper 0. Sorry, my cat is here. She just had a nightmare, I guess. So the T helper 0 cell is bathed by interleukin 12. So what will happen is it will become T helper 1. So, it is 12. So you take the first digit and that is a cell. And then this cell T helper 1 will release interleukin 2. That is the second digit of 12. So just a mnemonic to remember that interleukin 12 pathway makes T helper 1 cell, and T helper 1 cell would release interleukin 2. T helper 1 cell also releases interferon gamma. Now this interferon gamma will, normally we make this diagram like this as I have made here, that the arrow goes back towards the dendritic cell. But really this interferon gamma that is released within the lymph node, but it will also travel outside of the lymph node and go into the circulation and get spread in the body as well. So wherever there are dendritic cells or macrophages that are in the vicinity where they can receive interferon gamma, they can become activated. So interferon gamma's job is to activate macrophages and dendritic cells. So can you give me one quick second? My apologies. So my cats have Apple AirTag on them, and my wife was trying to find where Kyrie is. She's sitting here. Okay. So T helper 1 would release interferon gamma. Interferon gamma would activate macrophages and dendritic cells. And what does activation mean of a macrophage or a dendritic cell? Remember, dendritic cell and macrophages are part of the innate arm, and the T cells and the B cells are part of the acquired arm. Acquired arm activation, this activation actually takes anywhere from a week to 3 weeks. So macrophages got exposed to the infection on my finger the day the infection started. Then they became activated within 8-10 hours to 1 day. They then ran to the lymph node, and over there it took a week to 3 weeks for the T helper cells and T cells and cytotoxic T cells and B cells to become activated. Now those cells are releasing, T helper 0 cells are releasing interferon gamma. Interferon gamma in turn will amplify the innate arm and activate the macrophages. What is the difference of a macrophage that is active versus not? Was that macrophage at the site of infection not active? Was it not picking up the pathogens and working with them? Yes, it was. The difference in the activated versus non-activated macrophage is the following. An activated macrophage will accelerate phagocytosis. It can start eating very fast. A resting macrophage is still eating, but at a very slow rate. So why do we need to activate the macrophages and dendritic cell? Well, there is an enemy, a pathogen, that is growing fast as well. And we need our clearing cells, the cells that would clear the area, to work faster as well. So interferon gamma will activate macrophages and dendritic cell not only in the lymph node, but also where the infection was. And those macrophages and dendritic cells would over there become accelerated in their function and start eating up the pathogen quickly. This is called interleukin 12 interferon gamma axis. This is a hugely important bridge between the innate arm and the acquired arm, where they are amplifying each other. This is how we wind up the immune system to become better and faster and more active. So we use the word amplification. Both arms are now amplifying each other. Okay. So now the interleukin 2 is released by T helper 1. This interleukin 2 will act on cytotoxic T cells or CD8 positive T cells. These T cells are called effector cells. The T helper zeros are not called effector. T helper 1 are not called effector. Why are they not called effector? Because these are intermediate cells that are acting as a bridge between the macrophages or the innate arm and the cell that would do the final killing of the other cells, cytotoxic. So cytotoxic cell is called an effector cell. It does an effect. It would kill cancerous cells or virally infected cell or bacterially infected cell or fungal infected cells. So here imagine in this area, there are many cytotoxic T cells sitting there. One of them had the privilege to bind with an infected cell's MHC1. Cytotoxic T cells are CD8 positive, so they would bind with MHC1. MHC1 is expressed on standard cells, all body tissue nucleated cells. Now, remember that question, I forgot to answer this question. The student said the professional antigen presenting cells, they show MHC2. If they get infected, can they also present their infection on MHC1? Or are the professional antigen presenting cells only present antigens on MHC2? The answer is professional antigen presenting cells, when they pick up an enemy from outside and dismember it and show those pieces, that is on MHC2. But when a professional antigen presenting cell itself becomes infected, then it will present that on MHC1. That means macrophages, dendritic cells, B cells can present antigens on MHC2 and or on MHC1, depending upon if they are infected as well or not. So here we are just looking at some nucleated cell. For example, let's say it's a muscle cell or a skin cell where the infection was, or some lung cell where the viral infection occurred. And this cell is infected with, let's say, a virus. And this cell was presenting that, it was presenting the antigen, viral antigen on its MHC1. When this cell is doing that, then the cytotoxic T cell will bind to that area with its T cell receptor, Plus, if you see here, this is CD8 molecule that is bound with the MHC1. The important point to note is that there may be other cytotoxic T cells that are not bound to anything. So when the shower of interleukin 12 arrives, it will shower all of the T cells that are present, not only just a T cell, any other cell in the vicinity will be showered with this interleukin 12 or bathed in this interleukin 12. However, only those cytotoxic T cells that are bound to an infected cell will become activated. So that means there could be many. Imagine my room is an infected area and there are some cytotoxic T cells that are bound to some cells that are infected. And then the Kyrie that is sitting here, she's not connected with any cell. If you bathe us both with interleukin 2, if I am bound to an infected cell, I'll become activated. If Kyrie is not bound to an infected cell and she's also a cytotoxic T cell, she will not become activated. So this is a very important concept. So here, I made that concept as here is a cytotoxic T cell which is not bound anywhere. So it is just sitting there saying, oh, well, I'm not active. But this one is an infected cell. This is a cytotoxic T cell that is bound with it. Interleukin 2 will activate it. If you throw interleukin 2 on this free cell, it will not do anything. Yes. Beyond rocket science, yes. The answer is yes. So quick look at the comments. Okay. So now when this cytotoxic T cell has become activated, what it will do is that it will release two proteins that are important. And again, because there is this synapse, let me make it bigger. Because there is this synapse established between these two cells, in this synapse, the cytotoxic T cell will release perforins. Perforins are small molecules that can punch holes in the infected cell. Then another protein is released called granzyme. And I remember them, perforin is for perforation, causing holes, and granzyme is like hand grenades. So the other protein, hand grenades or granzymes, will then enter through those holes into the cell and initiate apoptosis or ubiquitination in the cell. They would try to kill the cell. And honestly, when a cell has developed holes in it, it has already become less integral. Things from outside would flow in and things from inside would flow out. And so this cell is already in jeopardy. So is this pathway clear? In this pathway, the effector cell, the cell that would do the final action, is a cytotoxic T cell. Now remember that the macrophages and dendritic cells are effector cells as well. In the beginning, they were picking up, they were sensing, they're surveilling cells, they were surveying the environment to pick up the pathogens or offending agents. But when they have interferon gamma released on them, they become active cells, and now they are effector cells. Now they are picking up damaged bacteria and the cells, etc. Okay, now let's stay in the lymph node. Let's go to the lymph node. So here is a dendritic cell. It's a different concept. Now we are talking about the B cell. So here is the dendritic cell that ran here from my finger to the local lymph node. And in that lymph node, once again, it bound to a T helper 0 cell. So if you see here, similar thing as before. So here is a T helper 0 cell. This is the T cell receptor that is bound to the MHC2 of the macrophage. The CD4 molecule from the T helper 0, remember T helper 0, CD4, binding with MHC2, multiplier is 8. So CD4 is now bound with MHC2. So this binding is done. The synapse is forming here. Then there are other molecules called PD1 and PDL that are also bound with each other. There are other molecules called CD28 and CD86 that are bound as well. The point is there are 4 or 5 arms that have to connect with each other to make sure that this activation is needed. This is called costimulation. However, in this case, on this T helper 0 cell, the bathing molecule is interleukin 4. interleukin 4 it's still a discussion. Where does it come from? There are various theories. We know that interleukin 2 comes from the macrophages and dendritic cell. For interleukin 4, we think that possibly mast cells can produce them. I believe some eosinophils can produce them. The T helper 0 cells, when they become T helper 2, that T helper 2 cell can produce interleukin 4 as well. But before it becomes T helper 2, where does this interleukin 4 comes from? It's an area of active research for years now. So I would just say this much, that when a dendritic cell in the lymph node has now bound with the T helper 0 cell, all the prerequisite bindings have occurred. Then from somewhere, if the interleukin 4 arrives, that interleukin 4 will transform this T helper 0 into T helper 2. So once again, this is not a separate cell. It is the same cell. In this place, it will become T helper 2. Just for illustration purposes, we draw it separately. So now it has become T helper 2. T helper 2 cell releases a number of cytokines: interleukin 13, interleukin 10, interleukin 4, interleukin 5, I believe even interleukin 3. I'm not sure about that, so don't quote me on it. So T helper 2 will now bind with a B cell. So we are in the lymph node. There are many B cells here. Once again, some of the B cells are bound to the pathogen. Which pathogen? The pathogen that from my tip of my finger, where there was infection, had floated in the lymphatic channel and reached this lymph node. So that pathogen here got captured by some B cell or B cells. There may be more B cells bound to it, but I'm just making one for clarity. What this B cell would do is that it would phagocytose it. B cells are professional antigen-presenting cells as well, in addition to making antibody cells. So they pick up the pathogen, they eat it up just like macrophages and dendritic cells, and then they present it on their MHC2 molecule. However, they present it for their own activation. Here, the macrophage or dendritic cell was presenting it for other cells' activation. It was activating T helper 0. Here, when the B cell presents MHC2 and binds with T helper 2, this presentation is for this B cell to become activated. It's a selfish behavior that B cell says, I'm going to present to you an antigen, but in return, I want to be activated. So when the T helper 2 cell will bind with it, and CD40 and CD40 ligand will bind together as well, the costimulation, the synapse is forming, then the T cell helper 2 cell would also release certain interleukins like interleukin 13, interleukin 10, 4, and 5. Especially the 4 and 5 are responsible for this B cell to become activated. And what does that mean? The B cell will be triggered to become proliferating, that is making more baby B cells, become differentiating so that it can make various kind of immunoglobulins. For example, interleukin 4's job is to trigger a B cell to make IgE. Similarly, interleukin 13's job is to trigger a B cell to make IgE. On the other hand, IgA. On the other hand, 4 and 5 also have an effect on the B cell of proliferating and differentiating or growing it. But remember, this interleukin 4 and 5 are activating a B cell after making this synapse. In the case of cytotoxic T cell, you did not need to have a synapse with T helper 1. T helper 1 just has to be active and release interleukin 2, and whatever T cell is bound to an infected cell will become active. Whatever is not bound will not become active. Here, a B cell has to be bound to a T helper 2 cell. And then when the T helper 2 cell would release interleukin 4 or 5, then this B cell will become activated and the B cell will become a plasma cell. The plasma cell will then start producing antibodies. So here there is a free B cell If you release interleukin 4 and 5 on it, this B cell is not bound to anything. This B cell will ignore those signals, will not do anything. This is a very important thing that many of the medical students ask me this question, that when we have interleukin 4s and 5s and 2s, why not all the cells in the lymph node become activated? They all do not become activated because they're all not bound to something. Whichever cells are bound and the synapses are produced, those cells will become activated. So Beyond Rocket Science says, maybe if you joined up with someone good at animation, we could use these in motion. Absolutely, that will be fun to see. Okay, so I hope, Margaret says, is the interleukin the same that it offset may become leukemia? The leukin was what I remember just thinking in writing. So leukemias are just the lymphocytes, the same lymphocytes becoming cancerous and making too many of them. Okay, so the leukin or leuco is from the white blood cells. So interleukins are the molecules that are exchanged between the white blood cells for their signaling. Okay, so I hope that this is clear. As much as this is a busy diagram, I have presented that diagram in a more traditional diagram that I used to make. So just a review of the previous one, but in a little more cleaner way. I hope you would declare this to be cleaner than this one. So here, this is the last part. Dendritic cell arrived in the lymph node. Dendritic cell itself releases interleukin 1, interleukin 6, interleukin 12, interleukin 23, tumor necrosis factor alpha, and beta, GM-CSF, and many other molecules. Interleukin 1's job is to go and cause fever plus activate the inflammatory system. Interleukin 6 job is to go and kick the liver to say, hey, start making acute phase proteins. Acute phase proteins by the liver are the proteins that are part of the innate arm, and that would help in inflammation as well. One very famous protein is C-reactive protein that is measured in the labs to understand if somebody has inflammatory state going on. But other than that, hepcidin, ferritin, and other complement proteins, etc., are produced by the liver as well when the interleukin 6 triggers the liver cells. Interleukin 6 also helps activate inflammation. Interleukin 6 also causes fever by going to the hypothalamus and changing our set temperature set point. Interleukin 12, you have already seen that interleukin 12 causes the T helper 0 to become T helper 1. Interleukin 23, I have not drawn it over here. It helps activate T helper 17 cells. Tumor necrosis factor alpha and others, we have discussed those in the past as well, related to inflammation. Okay, so if the T helper 0 cell is showered by interleukin 4, then it becomes T helper 2. It releases interleukin 13, interleukin 10, interleukin 4, interleukin 5. Interleukin 13 has an interesting behavior. One, it acts on a B cell to do a class switching, and class switching is a different topic. We'll discuss it at some other time, but just know that B cells can make IgM and IgG and IgE and IgA. So whichever antibody they're making, we say they class switched from a previous type to a different type. So interleukin 3 can act on a B cell and help it class switch towards IgE. Similarly, interleukin 4 can also act on a B cell to class switch it to IgE, plus interleukin 4 and 5 both help a B cell become differentiated and proliferated. Interleukin 5 acts on a B cell to class switch that to IgA. Interleukin 5 can actually also help activate eosinophils and other cells. Interleukin 10 is interesting. What it does is it acts on the innate arm and helps tame down, helps tone down the activity of the innate arm. It kind of reduces the activity of macrophages. It is anti-inflammatory. On the other hand, if you look at the brother of T helper 2, which is T helper 1, when T helper 1 becomes activated because of interleukin 12, it releases interferon gamma, and interferon gamma is a potent amplifier of the innate arm macrophages and dendritic cell. So T helper 2 reduces the action of macrophages and dendritic cell. interferon gamma amplifies their action. Now, T helper 2, the interleukin 4 that it produces, also can act on T helper 1s to tame them down. What does this mean? When a pathogen arrives in our body, it is possible that the immune system would activate both of those arms. But what happens is one arm will become prominent or dominant. So either humoral arm is the B cell antibody producing arm, or the cytotoxic arm, which is the CD8 positive cells that are killing the infected cells. Both arms become activated, but one becomes dominant. For example, 80% of the action is happening here on the B cell side and the 40% is happening here. So what happens is when one arm becomes dominant, it releases interleukins to kind of try to suppress the other arm. We do not want both arms to be active at the same time with the same potency. So interleukin 4 can actually cause T helper 1 cell suppression and suppress the cytotoxic side. Now, once the B cell, now remember I made this as a separate B cell with the arrow here. This is what I was showing here, that they are actually connected with each other. In the diagrams, in the illustrations, we make it like this, that T helper 2 will activate B cell into plasma cell. But no, they are actually bound to each other through the synapse. Then the T helper 2 releases interleukin 4 and 5, and the B cell becomes active. So here, this diagram kind of cheats here and does not present the actual synapse that is present. It just is an easier diagram to work with. So the B cell, when it becomes activated, it becomes a plasma cell. The plasma cell has a very unique nucleus under the microscope. Because plasma cell is an active cell, it is producing a lot of antibodies or proteins. Its genes are open, and those genes are continuously helping to make antibodies. Messenger RNA is being made from there and come out and make antibodies. This is why when you look at this B cell under a microscope, you would see that the nucleus looks like a sailor's wheel. And that is just an indication that this nucleus is active. The other interesting thing with the active B cell are they are like little pregnant cells. They have tiny baby immunoglobulins being formed in their plasma. So they take a more oval or egg-like structure because their plasma expands with the antibodies. In our body, the circulating proteins, for example, albumin or globulin or complement systems or hepcidins or ferritins, these are produced by liver. So liver is a huge contributor of the proteins that are circulating in us. The second biggest contributor of the proteins that are circulating in us are B cells. They make antibodies that are circulating in us, and antibodies are proteins as well. So of course, then B cells are busy making antibodies, and those antibodies will then go and bind with the pathogen and help clear those pathogens out. If we go this route now, so this is the humoral route. If we go this route, what happens is macrophage or dendritic cell presents the antigen to T helper 0 cell or naive T cell in the presence of interleukin 12, which comes from the macrophage or dendritic cell. The T helper 0 cell becomes T helper 1 cell. T helper 1 cell would release interferon gamma to activate the innate arm. It would also release interleukin 2. This binding, then there does not need to be a synapse here. No immune synapse needed here. Interleukin 2 is just released in the environment, and any cytotoxic T cell that is bound to an infected or cancerous cell through its MHC1 and MHC1 plus the, MHC1 is here, and T cell receptor is here, and CD8 molecule is here. So when this structure is formed and you pour interleukin 2 on them, this cytotoxic T cell will become totally angry and active. It would release perforins and granzymes that would kill this cell. Of course, these cytotoxic T cells release multiple cytokines as well that would then activate the neutrophils and other cells in the innate arm. I have also not make regulatory T cells. I have also not discussed how the class switching occurs in the B cells, but this is a general overview. And just one thing I left here, interleukin 13 that is released by T helper 2 cells can actually go back to the innate arm and change the behavior of the innate arm. Interleukin 10 suppresses the innate arm. Interferon gamma activates the innate arm. Interleukin 13 transforms a macrophage into the alternate macrophage. What is an alternate macrophage? It is an M2 macrophage, mostly declared as an anti-inflammatory macrophage, and this works more with the parasites and other type of pathogens. So as you can see here, the acquired arm has many interleukins that would actually affect the innate arm. And of course, innate arm has many interleukins that would affect the acquired arm. This is how these two arms work together. This is a brief, for 56 minutes, this is a brief overview of the immune system. What I have not discussed is how the immune cells are formed in the bone marrow. What I have not discussed is how the T cell get trained in the thymus, and what is positive selection and negative selection. What I have not discussed is that how the antibodies are formed, what is the structure of the antibody, what is the somatic hypermutation that happens there. Similarly, what happens to the T cell receptors, how are they formed and what is their structure. I have also not discussed how the class switching occurs from IgM to IgG to IgE to IgA. I have also not discussed how mast cells work and cause allergies. I have also not discussed how eosinophils, basophils, and neutrophils work, natural killer cells work. We can take that up at another time if you liked this lecture. So this is the discussion. Thank you very much for listening in. Like, subscribe, and share. There are links in the description of this video where you can become part of Patreon. We do 2 Zoom calls every Wednesday, one at 6 p.m. Pacific and one is at 1 p.m. Pacific, where we discuss these and more studies. So that is the discussion. I hope you liked it. Bob says, I always miss these live streams because YouTube doesn't notify me in a way. So many of my subscribers have said that we don't even get a notification. So that's just weird. Paul says, thanks, Mubin. Remember to like the button. Correct. Bob says, thank you for the lecture. Came in late. I'll watch tomorrow. Absolutely. And what else is happening? You're very welcome, Audrey. Bob says, from what I saw, this looks like a great lecture. Excellent. Good to know. Gail says, amazing discussion. I can't believe how much Dr. Mubin knows about just everything having to do with the body and how it works. The immune system is very complementary. Correct. So the important thing why I did this lecture is that in medical education, we understand these things piecemeal. For example, we'll understand how the B cells are activated, or how T helper becomes T helper 1 or 2, or how macrophages activate the cells. But there is no connected, integrated view of these. And when you follow the immunology books, they are so complicated that it becomes very difficult to connect these dots. So I thought I'll connect them for all of us here. Miguel says, this class accelerated my metabolism and losing weight. Thanks for the amazing review. You're very welcome. You're very welcome. Roller Girl says, I got the notification. Thank goodness. Thank you. And Andy says, oops, missed it. We'll watch now. Neepak says, great. Thank you very much. So thank you very much. Like, subscribe, and share. And actually put a comment in there. I think algorithm loves comments as well. So put a comment. There are links in the description to become part of DrBeen or Patreon or Substack. And I would see you tomorrow. Tomorrow, the lecture with Dr. Cyrus Kombucha is at 12:15 Pacific time instead of evening 6 o'clock. He could not come in at 6. If a magician says, thanks a lot, sir, for your you're very welcome. With this, thank you, and I would see some of you tomorrow. Bye for now.